Stay close to that. All right, great. So I'm just going to give y'all a quick update on research initiatives at COMBINEDBrain. And it's all based on my own personal experience. This is my son, Lou. That was Lou when he was diagnosed with Angelman syndrome 26 years ago. This is Lou now, getting an ASO. It took 23 years to get there, for him to get his first ASO treatment. He's 27 now. And my whole goal is to shorten that timeline for everybody else. So these are all the things that we do at COMBINEDBrain. And just now when Eric introduced me, he said, I started COMBINEDBrain, but I didn't start it by myself. Dr. Anna Falzer over there was my partner in crime from the very beginning, and I just want to make that very clear. So we try to do at COMBINEDBrain everything here in this whole pathway that needs to be done to shorten that timeline from diagnosis to treatment. And a lot of the stuff that we do is underground. It's not visible. It's foundational sort of resources. It's not necessarily basic science. It's not even necessarily translational science. It's putting together all the pieces that you need to be able to do that. So the very first step is diagnostics. And one of the problems with Angelman syndrome, and I actually don't remember, but I think it's the same with Kleefstra, is that the kids are not often diagnosed even now until they're over a year old. Usually they're over 2 years old. And so we we really tried to find diagnosed in the first year of life with. It's got its own website, projectfindout.org. And it's now, I'm going to show you a slide about pilot data, but we've gone beyond that. We now take insurance. And in fact, our diagnostic yield from the pilot data was 26%, which is really amazing. This is using this kind of vague algorithm, really. Babies under the age of 12 months, they have to have 2 of the symptom domains. You're going to think that they can't possibly be diagnostic. One of them is feeding difficulties. Every baby has feeding difficulties. Some of the other ones are hypotonia and other vague things. And yet, even with those vague diagnostic domains, we still have a 26% diagnostic rate for rare genetic disorders. So we do that, and please, if there's a baby that you think may have a neurodevelopmental disorder, send them our way. We're now getting a lot of referrals, believe it or not, from large medical centers, which drives me insane. The reason we're getting referrals is because even when they're in the NICU, the hospitals don't order whole genome sequencing because it takes away from the daily rate they can collect. You know what I'm talking about. And so they discharge them, and then they tell them that they will have their genetic testing at their outpatient visit with the geneticist, which they have to wait a year to get. So we are getting those referrals now. We also do a lot of collaborative preclinical research, and I have to say, IDefine has pushed us really hard on this, and so has Dr. Anna Falzer, to work hard to get a bunch of different groups together to be able to sort of try to be more efficient. So we might have 5 or 6 or 10 disorders all working together with one academic. So for example, you heard from Jeff Collar yesterday, and he talked about the poly-A mimetics, I believe. So we have a lot of disorders working through COMBINEDBrain with Jeff Collar to test poly-A mimetics across different disorders. We also are trying to come up with more efficient ways to do variant characterization. So instead of one-offs, for every single, new variant that comes up, and you have to find a professor who's got some money and got the resources to characterize the variant, we're trying to come up with more ways that we can do it more efficiently. And then high throughput screens and all different compound screens. And I think you heard from Unravel yesterday. And so we try to pool contracts for those screens too. We also have our biorepository, and this is another one that you're going to see Anna's picture on here. She helped start the biorepository. No, she did start the biorepository. And we now have, it is going quite strong. At this point, we have more than 40,000 tubes in the biorepository. It's at Van Andel in Michigan. We have more than 3,400 participants. We also collect a lot of controls. And we are sort of one of the few places where academics and industry can actually get a hold of controls. And so we're very proud of collecting our controls. We collected 1,000 participants last year, and 1,300 patients were consented last year. I think last year we had fewer than 20 roadshows. This year we've had 25 roadshows where we sent to a family conference to collect samples at the family conference. And it's sponsored by whatever disorder is holding the conference. But we are trying to work as a collective. And so whatever family conferences it is, any other combined brain disorder who lives in the area can also walk in with permission, of course, ahead of time, and do their biosample donation. So that makes it possible for us to have sites all over the country. That's how we've managed to collect so many. These are all the different kinds of samples that we collect, and they're in the biorepository, and they're available for all different kinds of researchers all over the world. We're sending samples constantly. We have data use agreements, material transfer agreements. We have a whole, one lady whose whole job is to deal with those DTAs and DUAs and MTAs. And we charge at cost, as close to cost as we can. So we really want everyone to take advantage of those. We are also doing biomarker development. And for biomarker development, we look at all different parts of the whole pathway. So genomics, transcriptomics, proteomics, and then we have to keep track of the symptoms, because otherwise, how are you going to know if the person is severely affected or mildly affected? So we have to keep track with validated surveys to be able to match the patient symptoms up to the omics. This kind of shows you a timeline of our omics studies. Right now, we're in the middle of a CZI-funded omics study. It's limited in that it's only 13 disorders, only 20 samples per disorder, but it's helpful because we have contracts for those, and other disorders can add on samples to those contracts at cost. So, that is exciting. And our next frontier is going to be longitudinal by hoping to be able to look at biomarkers across time instead of just a single point, so that we can try to figure out, you know, when things change during the lifespan, but also could be responsive to treatment. This tells you a little bit more about the Combiomics grant. It's called this crazy word because it's funded by CZI, which changed its name to BioHub with that bracket in the middle of the word. So we're just trying to be funny and do the same thing. And this is all of the data that we collect to go along with that omics study. So these are the options for add-on samples. So any academic researcher could go through the IDefine Foundation and talk to them about adding on additional assays. And so you can get them at a really low cost, and we can then be analyzing them in comparison with other disorders too, which gives you more statistical power. This shows you the Combiomics timeline, which we have just started this year, and we're about to submit our first batch. And then we'll be submitting another big batch next spring, and then hope to have all of our results returned by next fall and data analysis the following year. Natural history, we have to collect symptoms, we have to collect phenotype, we have to do that, otherwise we can't make sense of the biomarkers. And so we have a lot of caregiver entered data that we collect on Matrix, but we know that groups work with all these other platforms. For example, I think Kleefstra is on Citizen and on Simon's, right? Okay, so we try to not make people fill out the same surveys twice. It is really complicated, but we have managed to get data use agreements in almost every case. So we're able to tell people to go ahead and use whatever platform they want to, keep doing it. As long as it's a standardized, validated survey, we can align the data across data fields. These are the surveys we have on our portal, our Matrix portal. I don't like the seizure survey. We're looking for a better seizure survey. If anyone has any advice on that. I think the PELS seizure survey, which was recommended to us by the Childhood Neurology Foundation, I think it's great for clinicians in the clinic. I don't think it's a good measure of treatment effect. We need a seizure survey that's a better measure of treatment effect. The only thing we've been told to do is seizure counts, and there's no seizure count that everybody loves. So these are all the standardized, validated surveys we use. We picked them, and there's a big spreadsheet on our website that we update once a year with every outcome measure being used in any clinical trial for any of the 150 combined brain disorders, and we're up to almost 150 disorders now. So you can go in that giant spreadsheet and see which outcome measures are being used for which disorder, and that way you can actually pick the best outcome measures or the ones that you think the FDA likes the best. And so by doing that, that is how we pick these measures, except for the seizure survey, because we think that they are they're the most likely to be able to be used as baseline data in FDA applications. So data now on the Matrix that we can actually do cross-disorder comparisons. This is impossible to read. If anyone wants any help looking at this, I can send you this later. But basically from here, I can actually see it. And what I see is that, for example, muscles and bones, FAM 177A1 seems to have the worst issue with muscles and bones. And this purple one on the left is Schinzel-Gideon, seems to have the worst issue with digestive system issues. So we can actually look across a lot of disorders and try to figure out if someone has, for example, a compound that might help with digestive issues. We can suggest which disorders would be best for them to target, things like that. Here's another example, getting a little drilled down into the digestive issues. So you can see their feeding difficulties, defecation issues, dysmotility issues, and they really vary by disorder. So we're finally, now that we have enough, 3,300-ish or so patients in the database filling out the ClinGen surveys, we're able to really dig into it. Oh, we launched our EEG bank this year. In the Angelman world, we collected hundreds of EEGs, and anybody who knows about EEGs knows that Angelman EEGs are recognizable 500 yards away. And so you can diagnose a kid with Angelman syndrome based on their EEG pattern. And still, it took us hundreds of EEGs to do the right analysis to be able to use the quantitative pattern for a biomarker. So we want that possibility to be there for all of our combined brain disorders. So step one, piles and piles and piles of EEGs. And so we are doing that. We're collecting EEGs. EEGs can be retrospective, so anybody's EEG from any time in their life, they can upload onto our platform. We convert it into EDF format so that it can be analyzed by anybody. But we also collect prospectively now, and that is going great guns. We collect at conferences. The patient advocacy groups pay for it, but it's at cost. And those EEGs then go into the EEG bank. And we can collect an overnight EEG anywhere in the United States. We have IRB approval to collect overnight EEGs from the house, 18 hours, standard rig. Our industry advisory board and our EEG working group really specified exactly what we had to do that. And the texts that we use are the same texts that collect for Angelman. And so if the Angelman ones, they can do. And nobody's requested it yet. So please request it. It is a very good price. It is a very good protocol. It is available to you. I don't know if we're going to keep doing it forever if nobody wants it. Observational studies. You find a drug somewhere, I mean, maybe through Unravel, maybe through another screen, maybe olanzapine, whatever drug that you find, you can actually, if it's by a physician, the family can collect data ahead of time, during and after, in an observational way, and it's IRB approved to collect that data. We do not prescribe, we do not advise, we do not recommend. This is for families who are medications off-label or over-the-counter, and that then can be published as case series, or if there's enough of them, actually actually a paper. So what is an observational study? It is not a clinical trial. You have to talk to your doctor to get the right prescription or to be told what you should take. There's no placebo control. It's open label, but it is publishable preliminary data that you all can use for whatever you need. Okay, outcome measures. So how do we measure if those treatments are working? We have to have the right outcome measure. Okay. And so the outcome measures have to either be chosen from that FDA list, or we find that, in fact, there's a new outcome measure that needs to be created because there is no appropriate outcome measure. So in the way we figure that out is we do conceptual models. You all did one last year. It was fantastic. It was done here at Mass General. And so you look at the disease concept model, and that tells you the domains that are of concern to families. Those domains then are the ones that you're going to need to have an outcome measure for. So when we looked, so there are a couple different things you can do. One is we can do a global impression scale. Global impression scale, it could either be asking the clinician or asking the parent, how is your kid compared to other kids with the same disorder? How is the patient compared to other kids with the same disorder? So that would be a severity scale. Or it could be, how is your kid compared to last time? So that can be a measure of improvement. Global scales are handy because they can actually go across done scale. If it's not at all, I'm the most severely impaired. If it's improved, very much worse, we very much. And so we at COMBINEDBrain are working on creating this and doing it in a way, such a way that it could be used across all COMBINEDBrain on our matrix. And so that is actually a subject that is near and dear to a whole bunch of our clinicians and disorders and FDA. And so we're having, in fact, a research round sec. So when we started looking at domains needed outcome measures that did communication was a big one. So we worked on expanding the ORCA communication measure, which was actually developed for Angelman syndrome. And Bryce Reeve is the PI of that. Work with Bryce. And we have 12 disorders that are being studied for that. It's been under study for 5 years, and we're almost done. This year, it'll be published this coming spring, and hopefully what that means, and this has been an interactive project with the FDA. We meet with them every single month. We've gotten to know each other in such a way that we share recipes and talk about growing tomatoes, and I'm telling you, it's a lot more than just the outcome measure. It's really been a fantastic way to develop relationships with FDA. So, this is how the ORCA has been organized and funded. It's an FDA-funded project. Those are the 12 disorders we're expanding it across. They're wide enough in severity that we're hoping that we'll be able to use the ORCA across any neurodevelopmental disorder once it's finally published. But toileting, oh, I didn't know this was one of these kind. Let's see here. Okay. We found there was no measure for toileting. And toileting is a big deal. If a kid can be independent at toileting, that opens up a lot of opportunities for the kid and makes the family's life much better. My own kid at 27 years old has now taken himself to the toilet independently 3 times in the last month. I think he's developing that skill at 27 years old. That is life-changing. So we want to be able to measure. He doesn't get all of the steps perfectly. So we want to be able to measure how many of the steps does he get right. Is he moving up a scale? Can it be measured how much better he does? So we created a survey based on one that had already been used for Hunter syndrome but needed to be adapted to everybody else. Out to all of our combined brain disorders, and we had a bunch of kids from Kleefstra. Look, you're like number 9. We need more, but we did, SYNGAP blew everybody out of the water. But we did get a lot of data from that, which has been really helpful, and we see that the ORTA score, the scores go across all the score categories. There are a lot of scores in the 0 range. Those are kids that really are so severe they have absolutely no independent toileting abilities. We also measure normal controls, and we need more normal controls. Do you know a kid between the age of 1.5 and 4.5? We want to ask that person's family how well that kid is doing in toileting. We need more normal controls to be able to compare the ORTAs. So we are almost done, and we are ready. We will be ready this fall to release the survey to all researchers. It will be free or extremely low cost for academics. It will cost money for industry. Last but not least, well, second to last but not least, we've started a mortality study. This is an attempt anytime any child with a or adult with a neurogenetic disorder, a rare neurogenetic disorder, passes away, we want to collect data on why. We want to collect the medical record. We want to do get vital documents. We want to interview the family, and we want to interview the healthcare provider. And we're hoping to get enough over time. This is an open-ended funded enough to go on forever. We want people to be able to tell their stories, and for each disorder to be able to collect enough data to start to have a picture about what causes death. This is how it works. There's a caregiver intake survey. It's all done on our matrix. And then we do the health interviews, which are open-ended interviews, a lot like our conceptual model. And then finally, we also do this interaction with the FDA, and what we found is that this year we've started a brand new thing called the Research Roundtable for Neurodevelopment, and it is working with FDA and clinicians and patient advocacy groups and all different other stakeholders to try to make some kind of a decision on how we can measure treatment effect in these disorders. And the topic chosen by the working group this year was global scales. So that is what we were studying. We're doing that in September. And this is our current team. It's changing all the time, but these are the people there right now. And that is Combined Brain. Do you have any questions?